Good evening, everyone. I'm Namubudu. I'm the membership manager at Addenbrooke's and the Rosie Hospital. Thank you so much for joining us this evening for an interesting lecture on CAR T cells moving from cancer to autoimmunity. Without further ado, I'll now hand over to Chris White, our staff governor and host for this evening. Chris, over to you. Thank you.
Hello, good evening and welcome to everybody this evening. Thank you very much indeed to everybody joining. This is the most popular talk that we've ever had. So I'm absolutely thrilled that so many are turning up. A particular thanks to Dr. Ben Uttenthal and also Sarah Albon for presenting tonight. There will be a time for questions. There's an awful lot for us to go through this evening and we'll finish at 7:30. Okay, so I'm sure you're going to find this presentation extremely interesting. Cambridge is at the forefront of cell therapy treatment, and we all know how common cancer is, with around half of us getting cancer in our lifetimes, but also autoimmune disease, very common as well. So about 1 in 7 females that get autoimmune disease, and about half that, I understand, for men. So a very, very important subject. And as I said, we're right at the forefront on this. So Ben, if I went through all of Ben's biography, it would take the whole evening, along with Sarah's as well. We'll probably still be here tomorrow. In short, multiple degrees, studied all over the place, including the dark side, i.e. Oxford and Royal Free, Hammersmith, PhD, etc. But also very interesting over in the States, at one of the international centres there that had 3 Nobel laureates. So an expert in the field, and Sarah's role and training. So great, been at Great Ormond Street, University College London. A scientist that specialises in producing the products that are then used by Ben and the other clinicians to treat patients. So an absolutely critical role in terms of producing those world-leading therapies for our patients. So when you've got questions this evening, if you could possibly put them into the chat box. I think we're going to get an awful lot of questions. I'd like us to get through them all, but I very much doubt that we're going to be able to. What I'll try to do is to group them and then put them to my colleagues this evening. Okay, so without further ado, if I can pass over to Ben to start the presentation, followed by Sarah. Thank you very much.
Thanks very much indeed, Chris, and thank you for the kind introduction and for the opportunity to talk about what is obviously an area very close to my heart. I'm just going to see if I can share my screen, so bear with me while I do that, and tell me if you can see a screen saying CAR T cells moving from cancer to autoimmunity. Brilliant.
Yeah, that's fine.
Perfect. So this is our title. CAR T cells lend themselves to dismal puns about moving and transport and things. So in this case, we're moving from cancer to autoimmunity because CAR T cells have been revolutionary in the treatment of some types of cancer and are now increasingly being applied to other areas of disease, including autoimmune diseases. So I'm going to talk a lot about cancer and then talk a little bit more about how things are developing in the field of autoimmunity. It's not a new thing that the immune system has been used to treat cancer. And in fact, the first attempts at doing so go all the way back to the end of the 18th century, when the surgeon to the Duke of Kent, who was relatively up to the minute in his reading of the medical literature, had heard about the success of vaccination for smallpox by injecting people with cowpox, and wondered if he could do the same for cancer. So the exact method is not recorded, but it's thought that he took one of his patients, got some of their cancer cells, and tried injecting them into himself. Sadly, we don't know what happened, but we don't think it actually would have worked to help him stop getting cancer. But it was the idea that was important, and the idea of stimulating the immune system to stop or treat cancer were taken forward perhaps most successfully in the first instance by someone else called William Coley, who back in the 1890s used some heat inactivated bacteria to try to treat patients with cancer. So what he did was he got some streptococcus bacteria that would usually make people feel very unwell with infection. He inactivated them so they couldn't actually cause infection, but they could still make people feel pretty unwell when they were injected, because when they were injected, they stimulated the immune system. And remarkably, he was able to cure a small proportion of people with otherwise incurable soft tissue sarcomas. And that, again, showed how you could use the immune system to treat cancer. And then jumping forward to the last century, increasingly people began to understand how that could be used. And in the 50s, the first donor stem cell transplant were carried out using the power of a donor immune system to try to treat a cancer that a patient's own immune system had not been able to recognize and attack with sufficient ferocity to stop it turning into cancer. And we still, of course, use bone marrow transplant today for that purpose, although we got more sophisticated in how we do them. And then if you look towards the 70s, 80s, 90s, we began to understand more about how the immune system and the immune response to cancer worked. And that led to, in 2017, the first instance of what's called a CAR T cell, which I'm going to talk about it in great detail, being approved to treat a blood cancer, leukemia. And the NHS was one of the first jurisdictions worldwide to jump on this. So it was only a year later it got introduced into the NHS with a great fanfare, and we've been doing it here in Cambridge now since 2020. And as things have advanced, these therapies started to be used for blood cancers, but are now being used for solid tumours. And we know that there are now some similar approved cellular therapies called tumor infiltrating lymphocytes, or TILs, being used in melanoma. So that's what's happening in cancer. It's even more recent that they've begun to be used for autoimmune disease. And just a couple of years ago, CAR T cells have begun to be used for systemic lupus erythematosus, what we often call lupus, and that is causing great excitement in the field. So what is a CAR T cell? Well, the most commonly used type of CAR T cell, the so-called autologous CAR T cells, that means taking a patient's own blood cells. And of course, the cells that we're interested in are the immune cells, or specifically the T cells. And these T cells are ones that are usually involved in causing all kinds of different immune responses against loads of different kinds of infections, and indeed cancer cells should they arise. So there are thousands and thousands of different T cells in the body, but they all attack different things. So the theory here is that if instead of them all attacking different things, they could all be genetically modified so that they all attack the same thing, in other words, the patient's cancer, then they could be really helpful. And that's what we can do by turning them from T cells into CAR T cells. And what we do with when we make CAR T cells is to use a viral vector that modifies genetically that T cell and inserts the gene for what's called a chimeric antigen receptor, or CAR. And that chimeric antigen receptor we'll come on to in the next slide, but that's what targets a T cell so that it goes around looking for and attacking what you want it to attack, the thing that is bound by that CAR, that chimeric antigen receptor. So you modify those T cells into CAR T cells, you grow them up into millions of cells, all the same, in the laboratory. And then usually after giving the patient a small amount of chemotherapy to make room in the immune system that the cells can then grow into, you infuse the CAR T cells into a patient just like a blood transfusion, and they drip in and they start going around the body looking for their target to attack. And they will rapidly find their cancer using the chimeric antigen receptor on their surface to find it, and then they'll start to attack it. And through this immune response against the cancer, it will be a really strong response to attack cancer in a different way from the way that we usually use in chemotherapy or radiotherapy. It uses those CAR T cells to attack and kill cancer cells, and it is revolutionary in some blood cancers. It is now beginning to be revolutionary in some other types of cancer. So how does the chimeric antigen receptor work? Well, here's a sort of scientific diagram of what a chimeric antigen receptor looks like. And you can see that the CAR on the right of the screen is made up of 3 different domains. And perhaps one of the most important domains, although they're all important, is this one at the top. It's the bit that is called a single chain variable fragment, or scFv in scientific jargon. And it's taken from a specific antibody. And you can therefore take it from an antibody that binds whatever you want. That's what means that you can target anything at all that's on the cell surface. So you get the bit of an antibody that binds its target, and you use that at the binding part of a chimeric antigen receptor. Then in the middle, you've got a hinge domain, which just allows the binding part to flop around enough so it can get oriented and bind properly and comfortably. And then in the middle, of the cell, this green and yellow stripy bit is an important bit, which is what does the signaling. So that's a bit that when the outer binding bit of the chimeric antigen receptor has attached to its target, then it activates the signaling domain and tells the T cell that it's got to get active and start looking around for something to attack and kill. So the picture I've just shown you is from the first generation of CARs, and you can see that second from the left in this slide. Relatively simple. It's got this binding domain here shown with the kind of green lozenges, the hinge domain in the middle, and then the signaling domain. And in fact, when they first started using those CARs, they found they didn't really work very well. What has really made the difference is the introduction of one more refinement to this initial CAR concept. And you can see here in the second generation CARs that they introduced a separate molecule called a co-stimulator molecule, and the common ones that are used are CD28 or 41BB, and they're co-stimulatory molecules that give the T cell a little bit of an additional kick, so that when it binds through the CAR, then it's already co-stimulated, and that makes it work really well. So these second generation CARs are the ones that have been used in most of the studies and are now used commonly in the NHS today. We use them routinely every week to treat patients. And as some people are trying to make CARs more and better, they're looking at other ways of getting them to work more effectively. And the third generation of CARs uses a relatively simple concept of taking both the co-stimulatory domain CD28 and 41B and putting them into the same CAR. Not yet known whether or not that actually helps them to work better. And then many different laboratories around the world are looking at fourth, fifth, sixth subsequent generation of CAR, and they're called quite fanciful things. If you're trying to make the CAR better, you can call it an armored CAR, and one way of armoring your CAR is to get it to secrete other molecules like interleukin 12, a kind of immune system stimulating molecule, so that when the CAR binds, also it helps to harness lots of other immune responses by secreting this IL-12 molecule. And then I won't talk much about this, but it's a clever thing on the right, what's called a switch receptor, that stops the CAR T cell being exhausted by constant exposure to cancer cells, and actually means that what would be an exhaustion signal is turned cleverly into a further stimulating signal to give that CAR T cell an extra boost. It's like an arms race between the cancer tumor and the CAR T cells, and this is one of the ways in which CAR T cells might be able to win. So lots of different ways of making CAR T cells, but there's no point in making a CAR T cell unless you've got something good to target against. And the cancers that have really set the trend for CAR T cell being used are the B cell blood cancers, because it turns out that if you direct CAR T cells against B cells, then you can kill them really effectively, and actually many patients can get away without their normal B cells quite well. Now, at the top of this slide here, you can see the normal development of B cells, all the way from a stem cell through a primitive B cell up to what we call a plasma cell, which is the cell that usually functions to make lots of antibodies against a particular type of infection. And if any of those stages go wrong, they could form a cancer. So if a plasma cell goes wrong and becomes cancerous, it causes myeloma. If a pro-B or pre-B cell goes wrong, it causes a type of acute lymphoblastic leukemia, and so on. Now, the thing that all of these B cells have in common is that they all express on their surface a molecule called CD19, and it means that CD19 is a particularly good target if you're trying to make a CAR T cell that attacks different kinds of B cell cancer. And so the ones that have been most successful are CAR T cells directed against CD19. And I'm just going to flick through this slide quickly, and I don't really expect people to read it, but there are all these names in bold on the left, sort of rather unwieldy names called things like tisagen, electrocell, and axicabtagene, silolucel, and so on, which I've been practicing, but really the drug company wants us to call them by their proprietary name of Kymriah and Yescarta, which is easier to pronounce. Anyway, you can see that these CAR T cells are reaching use not only in the NHS but in Cambridge, and in Cambridge in these various years we started to introduce more and more different kinds of CAR T cell against more and more different kinds of blood cancer, and all these things ending in L at the end, things like DLBCL, TFL, PNBCL, they're all different kinds of lymphoma, or some of them are leukemia, and we're using them all to treat patients with those kind of conditions here in Cambridge. Do they work? Well, this graph is a way of showing that yes, they do. So this is for for aggressive B-cell lymphoma, and if you look at this graph, it shows the time going on the x-axis, and on the y-axis it shows how many patients are still alive. And you can see that in the red line looks at the state-of-the-art chemotherapy before CAR T cells. And unfortunately, as time goes on, you can see that from 100% of people being alive, more and more people are dying, and that survival curve is going down and down until only about 20% of people are still alive in the long term. Whereas you compare that with Zuma-1, which uses the most commonly used CAR T cell for this kind of lymphoma called axicell, and we know that you can see that more than twice as many people are surviving long term. And this paper was not a direct comparison. It was done in what's called a propensity balance scoring analysis, because that's one of the best ways of doing these comparisons. And it shows that we're getting significantly better results with CAR T cells than we were with all of the best available treatments beforehand. One of the reasons for that is that CAR T cells don't work like chemotherapy, which leaves the body very rapidly, usually being excreted by the liver and kidneys shortly after you've given it. But CAR T cells are a living therapy, and they stay in the body, continuing to have an effect against B cells for many months or even years. And this complicated slide I put in to make the single point that patients may not be in what's called a complete response CR immediately. They may be in only a partial response PR. You can see in the middle. And the bit of this slide I want to point out is that if you look at month 1, there are quite a few patients in grey who only have a partial response. And then you can track those patients with the lines going towards the right, and you can see that some of them, not all of them, but some of them move up such that by month 3 or month 6, they are going into the complete response domain. The patients in grey are becoming patients in blue, and that's because those CAR T cells are continuing to have an ongoing effect, munching away at any cancer cells as they arise. And it's for that reason that CAR T cells can be more effective than some of the other more traditional treatments for cancer. So that's some of the good things about cancer, about CAR T cells, but no talk about CAR T cells would be complete without mentioning that the currently used CAR T cells are very strong, they're potent, and they do have side effects. And this slide is an old slide. It's full of small writing, but it gives you an idea how many different side effects CAR T cell can have. And the reason they have side effects is that they when they get activated, they release lots of different immune chemicals that help to generate their immune response. But those chemicals are the same ones that help to generate a response against infection. And so when CAR T cells are in a patient being activated by their cancer and having their response against the cancer, they release these chemicals called cytokines that make people feel just as they would if they had an infection, and sometimes as they would if they had a pretty bad infection. And so that can make people feel very unwell, and occasionally they might even need to be supported in the intensive care unit. They can have all these problems with low blood pressure and cardiovascular problems, highlighted in the top right. And sometimes these circulating chemicals can inflame the brain and mean that people have difficulty concentrating or become drowsy, and cause neurological problems. Now, fortunately, we have antidotes against these side effects to damp down the unwanted excessive immune response, but we still need to use them in quite a number of patients. And one of the things we want to make better about CAR T cells as we develop them going forward. dealing with these side effects is something that we've got better at over time, and that's what these graphs show. Now you're getting used to looking at survival curves, and these show in blue UK data of when people in the UK first started to use CAR T cells, and you can see now that relatively few people survived. In the right-hand graph, only about a third of people survived long-term, and that's partly because we weren't so good at selecting the patient to give them to and knowing how to support them from the side effects. But if you look at the blue and red curves, which are later data from 2020, which is when we started in Cambridge, and 2021 to 22, people are getting better at it, and it shows that we're learning from experience about how to deal with side effects and make CAR T cells work better. Despite that, there's still a lot of variation between centres in the UK, and these are the only data that are available that show how different UK centres have different survival. So each of these curves represents the data from one different UK centre in the earlier days of CAR T. And you can see that some patients in some centres did better than patients from others. And of course, I wouldn't show this slide if I wasn't able to say that we're in Cambridge, the top curve. But although I'm absolutely delighted by that and very proud of those outcomes, I'm fully aware that as we do more patients, we're likely just to revert to the statistical mean. But at the moment, we're doing well, and we're pleased with that. So what are we doing in Cambridge? Well, on the left-hand side of the graph, you can see a number of trials and things we're doing and building up. And I'm not going to talk about that now because we'll mention a lot of them later in the talk. But I'd like you just to have to look at the right side of the graph, which shows Cambridge, and it shows all of the other CAR T centres that were commissioned by the NHS in England to deliver CAR T cells in the first few years of CAR T cell therapy being available. And you can see that there's a big empty area around Cambridge, which means that we provide CAR T cells for quite a large region of the UK. And I've just added there the most recently commissioned alternative CAR T centres in Nottingham, Sheffield, Oxford, Southampton. And even with those new centres, Cambridge still has quite a big area that it serves, and it means that we're able to help a lot of patients from across a wide region. And it also means that if we want to develop things to new CAR T cells to deliver in clinical trials, and we hope advance the field further, then we can offer them to patients from across a wide region of the UK. There are a lot of developments going on in the CAR T cell therapy space and in cellular immunotherapy in general. And this slide is one that was first published back in 2019, and it shows all of these different commercially developed products. Each of the icons shows which company is developing a new product. It represents one product from one pharmaceutical company. And around the edge of the circle here are the products in early phase development back in 2019. Then as you work towards the middle of the circle, you can see they're going through early phase trials, phase 1, phase 2 trials. Then if they go further through development, they go through pivotal trials, and eventually right in the middle, they've been marketed and are being widely used on a regular basis. And you can see in 2019, the only two products that were available, Yescarta or Kymriah, the two products that I mentioned that we use commonly. How are things changing? Well, of course, all of these things that were out around the outside of the circle in 2019 are now marching towards the middle of the circle. And I just put in a few examples there of things that have now more recently become licensed. So Lysocell now reached the middle of the circle. That's going to be funded in the UK, we think, in this quarter. We're expecting to start using that very soon. Lifileucel, the tumor infiltrating lymphocyte product I noticed, has gone all the way from some of the first trials that we actually ran here in Cambridge now into the middle, and we're talking about using that routinely in the UK and so on. We're beginning to use the products in different kinds of lymphoma, in myeloma, and even they're beginning to show successful results in solid tumours like gastrointestinal cancers. That research is not just going on elsewhere in the world, it's also going on in Cambridge. Here's some of the conditions that we've been treating in trials here, and you can see they range from blood cancers like leukemia, lymphoma, myeloma, to solid tumours like melanoma, lung cancer, germ cell tumours, including CAR T cells in germ cell tumours. So we're setting up and these trials, and in many of them we've already successfully treated patients. So that's the way the field is heading, but it's got a long way still to go. And particularly if you use CAR T cells to treat solid cancers, there are some unanswered questions. You need a good target, and if you look at the top left, section A on this slide, you can see that if you've got a target that is expressed on your cancer, but happens also to be expressed at low levels on normal cells, well, CAR T cells are so good at recognizing targets that they might also start to attack your normal cells, causing problems. So that's called on-target, off-tumor toxicity, and that's a major reason for needing to have a really good target for CAR T cells. That's been one of the blocks to rolling out CAR T cells into solid cancers. Another problem is that solid cancers have lots of different types of cells all buried within one tumor. They don't all express the same targets, and so you need a good target that is expressed on as many different cells within the tumor as possible. If you look at in the bottom left at C, the CAR T cells have actually got to get into the tumor in the first place, and that's not always straightforward. It's one of the things, one of the ways in which you can armor your CAR T cells is to make them get better at trafficking to the tumor. And then when they've got into the tumor in D in the bottom right, they've got to resist all of those fiendish mechanisms that tumors have to try and damp down immune attack. One of them I mentioned is that by making by making CAR T cells exhausted. That's called a PD-L1, PD-1 axis, and that way of armoring the CAR is a way of overcoming it. But there are lots of different sneaky tricks that tumors have, and so we need to work out how we can best engineer our CAR T cells to overcome them. So there's plenty of work to be done, and that's one of the reasons why relatively recently in Cambridge we set up this cancer immunology program. That's led by me and Mike Chapman, who's the academic lead, and we have a lot of help from our managers Huiling Wu and Claire Goodchild. It's a relatively recent program, and we focused it because it's got relatively little funding as a new program on trying to develop new cell therapies, and specifically new CAR T cells against new different kinds of cancer. And to do that, we brought together 25 different pre-existing research groups from across lots of different organizations, 21 different organizations in the campus, to bring together people with this common aim of trying to develop CAR T cells against different kinds of cancer. And to do that, we've harnessed a lot of pre-existing expertise. There are many different groups working on lots of different solid cancers and blood cancers, and as a result we already have access to a lot of different tissue banks. Mike Chapman's expertise is in many things, but particularly in identifying targets on the surface of cancer cells. He has particular expertise in what's called proteomics, which looks at the cell surface proteins, and in the bioinformatics to interpret the enormous amount of data that you get when you work out all of the many thousands of different proteins that are being expressed on different cells. And he can use his skills in bioinformatics and proteomics to develop to work out very specific targets that would be good ones to attack, either with things like antibody therapies, I put them here, monoclonal and bispecific antibodies, or of particular interest to us, CAR T cells. So if you identify the target, the next thing is to work out something that can bind that target specifically, and that's what you use to form the binding bit of your CAR that I showed you right at the beginning. And you can do that with this VHH library, which is a technical way of making things that can bind anything. And again, Mike's group have developed the VHH library, which is available for collaboration across anyone on the biomedical campus to develop binding molecules against their target. So the next steps will then be to take that through manufacturing in a very precise way, such that they can then be used in clinical trials for patients. The things with ticks are what we've got available on campus, and Sarah are going to talk a lot more about how we use the new facility to do the final bit that we need to manufacture for clinical trials. The Cancer Research Hospital is certainly going to accelerate that, and of course we're delighted only a couple of days ago to have been given final formal go-ahead by the government that that's going to be happening. So I've spoken a lot about cancer. I've just got a few slides that we will need to hear about some of the things that we can do with CAR T cells beyond cancer. And we're going to talk a little bit about autoimmunity. Again, there's been a lot of excitement in this. Some of the major drug companies like AstraZeneca here in Cambridge has announced some tie-ups to start attacking autoimmune disease with CAR T cells, in fact, with what are called regulatory T cells. So these work in many of the same ways as the effector T cell, but when they are stimulated through their CAR, they act to damp down an unwanted immune response. And that means that, in theory, you could damp down an unwanted immune response against whatever you like. And we're doing a trial with them to damp down unwanted immune responses against transplanted livers, so that the hope is that people might be able to stop what would otherwise be lifelong immune suppression after their liver transplant. So that's one trial we're doing here in Cambridge, but we're also doing other trials in autoimmunity that I'll come on to. And there's a lot of excitement in the field about how you might even be able to engineer CAR T cells to target scarring fibrosis in the heart, so that it can be used potentially to help make people's hearts better after heart scarring diseases such as heart attacks. So far, that's only been used in animal experiments, but there's a lot of excitement about using them in humans. So how can we use CAR T cells in autoimmune disease? Well, you'll know that autoimmune disease is often mediated by unwanted antibodies. And as we said at the beginning, antibodies come from B cells, often plasma cells or other B cells. So the same CAR T cells that we could use to attack cancer B cells can also be used to attack B cells that are not cancer, but they're still making antibodies against things you don't want that are causing autoimmune diseases. And so that's what this slide shows. You can see on the right-hand side of the graph that you can attack autoimmune diseases that are caused by B cells, such as lupus, which makes all kinds of antibodies that can cause problems, or B cells that cause more specific problems like myasthenia gravis, where you have a particular antibody against the neuromuscular junction that makes people feel really, really weak. So you can choose to be more or less specific about how you use CAR T cells, and you can either target them to a specific B cell that is just making your bad antibody, or you can be less subtle about it and just wipe out all B cells, including the ones that might cause lupus. And that second approach is being used with some success. And here's one of the recent publications about it, and there's a lot of excitement about this because these patients have been treated often with other therapies to attack B cells, but they've still been very poorly. And what we found is that if you attack B cells using CAR T cells, the CAR T cells reach the B cells that the other treatments couldn't reach. It gets rid of them very thoroughly. And that means that patients were suddenly being, for the first time, put into complete remission from their autoimmune disease. These graphs tend to show that, try to show that in a rather unexciting way. What these graphs show is that they've got lots of different antibodies against things like double-stranded DNA and single-stranded DNA and lots of other things. And you can see that on the left-hand side of each graph, they've got higher levels of antibody, and then after they've had the CAR T cells in the black dots, all of the levels have gone down, and that's why patients began to feel better. Summarizing how the patient actually felt is more difficult, but these are scores of disease activity. So if you look at the top left-hand graph, a SLEDAI score is something that goes up in patients who have SLE. And you can see that all of the scores went down with time, showing that these patients felt dramatically better after their CAR T cells. And it was the same with the sort of equivalent scores in inflammatory muscle disorders, IIM, in the middle panel, and in systemic sclerosis on the right-hand panel. Everyone was in these series was feeling better. And then a final set of very scientific-looking naive graphs which show that actually, to people's surprise, let's perhaps focus particularly on the pre-switched memory B cell graph in the third from the left. So the memory B cells went down immediately after the CAR T cell had been given, but they then began to recover. So they begin to go up again, and despite that, which was a good thing, meaning that normal B cells were beginning to recover, they found that the patients still remained in remission. So what we think is happening in diseases like lupus is that the CAR T cells are getting deep into the tissues, getting rid of all of the B cells, but then there's a B cell reset, and some naive normal B cells are coming back, enabling patients still to produce antibodies against infection, but not to have lupus. We're doing studies in lupus in Cambridge, and this is the trial that in fact, I'm going to see the first patient tomorrow in clinic to treat in this first in human trial for CAR T and lupus. We're looking forward to running that. And as we begin to do more and more of those type of studies, we're going to need increased clinical facilities in which to do them. And so in my last couple of slides, I'm just going to show you our vision to create a new clinical space staffed by nurses and doctors who are able to deal with therapies of this complexity and look after patients with all of these different kind of problems, autoimmune and otherwise, that could be treated with cellular immunotherapy. We're going to call that the Advanced Clinical Immunotherapy Unit. We have a footprint for it, but we don't yet have the money. And so if you happen to be someone listening with very deep pockets who fancy putting a name on a room or an Advanced Clinical Immunotherapy Unit, then please do get in touch. We hope in following years to build on that unit to develop an Advanced Immunotherapy Institute with all of the things that bring together laboratory research through teaching and dissemination of expertise and manufacturing into treating patients in the clinic. So with that, I'm going to finish on the clinical part of the talk, and I'm delighted to hand over to my colleague Sarah Alban, who, as director of the Cell Therapy Lab, is instrumental in actually making these things happen. Sarah, if you say next slide, I can move them on for you.
Thank you so much, and thank you to everyone for skipping your dinner and attending. I feel very honoured and privileged to be able to speak to you this evening. So my name is Sarah Albert, and I'm the director of the Cambridge Cellular Therapy Laboratory here in Cambridge. And my talk is very, very short, and it doesn't contain nearly as much exciting clinical things as Ben did. But what I'd like to do is really showcase to you the laboratory side of making cell therapy treatment in Cambridge happen, a little bit about the laboratory's history, what we're doing right now, and then what we plan to do next. So thank you, Ben. Next slide, please. So this is another little timeline, but focuses on cell therapies at Cambridge. So the first BMT unit, so the first bone marrow transplantation unit in Cambridge, was established in 87, with the first autologous harvest happening in 87, and then the first allogeneic harvest happening in 1990. So the bread and butter of cell therapy very much started out as stem cell transplants and has since then exponentially diversified out. So after 1987 up to about 2008, there was a steady increase in workload, lots more patients wanting the service. We had various name changes, and in 2018, finally settled on the name, the Cambridge Cellular Therapy Laboratory, to really encompass the huge variety of cell therapies that were coming on the scene. It's not just about stem cells anymore, and our lab was needed to handle them all. Something very exciting happened in 2018, 2019, and that was when the first cellular advanced therapy trials happened at Cambridge. Ben's already touched on this before, and they were the tumour infiltrating lymphocyte cells against solid cancers. So it was really good. This was Cambridge sort of dipping its toe into the advanced therapy trials. And then everything kind of went a bit crazy from there, and it's been amazing to be a part of it. So in 2019 was when I came on the scene from spending 10 years in manufacturing cell therapies at Great Ormond Street and University College London. After a lot of lobbying, I managed to achieve some really significant infrastructure funds for the department. We had a huge shift in senior management, and it was a really exciting time because we also, at this point, became part of the second wave of NHS commissioned cell therapy CAR T studies, commissioned products, sorry. And we also got involved in a lot more clinical trials. So we're in 2025 now. We're incredibly busy. We've been through an awful lot, but we're looking towards the future with two main goals. Firstly, to really diversify the cell therapy service that we currently offer, but also really embrace the opportunity to be able to manufacture our own cell therapies on site, so they're much closer to the patient. Next slide, please. So a little bit of a background. Again, we are, as a department, officially part of clinical haematology in Addenbrooke's Hospital. We're part of the Cancer Directorate, although I think Ben and I have both sort of made it clear that cell therapy is not just about cancer. It is really branching across many, many disciplines now, and the need is only going to get greater. We are physically located in the NHSBT Cambridge Blood Donor Centre, which I always say is bang opposite Car Park 1, because everybody seems to know where Car Park 1 is. So opposite the entrance to Car Park 1, that's where we are. We've got our very own 3 clean room facility and some small wet lab space. And I think I just wanted to highlight here that the fact that we're on the Cambridge Biomedical Campus is really an incredible opportunity that needs to be embraced. We're the largest biomedical research campus in Europe. There's a hugely intense hotspot of research, plus a big hub of clinical activity with the 2 flagship trusts we've got here as well. We've got exceptionally close links with the Cambridge Medical School, as well as world-class research institutes. So we are very, very well placed to really embrace the cutting edge research and translate that into clinical therapies for our patient cohorts. Next slide, please. So some pictures of faces for you. My senior management team consists of Claire Gilby, an incredible operations manager for the whole of the Cancer Directorate, and has helped me keep the department afloat for the past 5 years. Charles Crawley is the director of the bone marrow transplant service for the East of England, and is also a consultant transplant haematologist. I'm the laboratory director, and then for the past year I've been working with Dan Gleghorn, who's joined me as an associate operations manager, which was a new role developed specifically because the service was exploding at such a high rate. And although not strictly part of the day-to-day running of the facility, we do work very, very closely with Ben, who is the lead for the cell therapy ATIMS across the trust. And I work closely with Ben to sort of direct the service in terms of what areas we're going to focus on and to build up the cell therapy portfolio. So we've done an awful lot of recruitment over the past year, and we're now at full establishment with about 29 members of staff, I believe. We've got a skill mix that's quite varied. We've got senior managers, we have quality personnel, production scientists, registered healthcare scientists, and medical laboratory assistants. So we're a big team. What exactly do we do? So the very heart of our service is supporting the East of England regional bone marrow transplant programme. So we look after the whole of the East of England, with patients being directed from Norfolk and Norwich and the East Suffolk and North Essex NHS Foundation Trust as well, which is primarily Ipswich. And in a nutshell, if someone asks me what CCTL do, I say we operate as a regulated cell handler. So any cells that are coming in and out of the hospital, they come via us, and we make sure that they are handled within the right legal and regulatory guidelines that govern our area. So we facilitate the procuring of cells from patients and donors, and we do this in partnership with other departments in the hospital, such as apheresis and surgery. We sample cells for testing. We do test some of them ourselves. We process cells, and that mainly consists of cryopreserving cells, so they can be stored in liquid nitrogen for long periods of time until patients need them. We store cells up until about 10 years, and then we start asking clinicians and patients if we can sort of start making some room. And then we issue cells to patients for their therapies. So there's a lot of governance, as you can imagine, surrounding that, and that's why I describe us as a regulated cell handler. So we have an adult service and provide a paediatric service as well. We facilitate allo transplants, which is when cells are harvested from another human, either a sibling or an unrelated donor. For transplant, and also we offer an autologous service as well, which is where cells are procured from the patient themselves. We then cryopreserve them and store them whilst the patient's undergoing their chemotherapy, and then the cells are reinfused when the patient's ready to grow a new immune system. Various ice cream flavors of transplant. The main one is apheresis, which is where we collect peripheral blood donor stem cells before processing. We also offer cord transplants, although we don't harvest the cords ourselves here. We receive them in from registries from all over the world. And we also facilitate bone marrow transplants as well, and participate in the surgery to procure marrow and make sure it's handled correctly. Another thing we do is donor lymphocyte infusions, which can be given as a top-up to patients that have already undergone a transplant. A few numbers from 2024. Sorry, Ben, you're a little bit quick. Is that the bulk of our work is the autologous transplants, closely followed by the allogeneic transplants. But yeah, approximately 150 odd patients a year, and that's steadily increasing. Next slide, please. So what else do we do? So Ben's already outlined that we are a commission centre for CAR-Ts. We have the same role to play here in the laboratory, where we operate as a regulated cell handler. And in the past, since we opened in 2020, we've treated in excess of 100 patients with one of the 3 main commercially available products. I won't try and pronounce the drug names. I just call them by the brand names. So Yescarta, Tecartus, Kymriah. That's a big enough mouthful for me. And in this setting, we're responsible for the procuring of the cells, the receiving, testing, processing and storing, and then we make sure that it's shipped out to the right manufacturer somewhere else in the world for the CAR-Ts to be manufactured. And then the product comes back to us, we receive it, store it, and then make sure it gets to the right patient. Next slide, please. And this is one slide just to say it's not just about CAR-T, it's not just about cancer. Another cell therapy that we've started facilitating recently is for patients with cartilage defects in their knees. So in this situation, we facilitate the procuring of cartilage biopsies from patients' knees in surgery. The cartilage is then sent off to a company in Germany for manufacture into chondrocyte building blocks called chondrospheres, and then they can be injected back into the patient knees to build new cartilage. So this is just another example of how diverse cell therapy is, and that there really is so many applications of it. Next slide, please. And if that wasn't enough, we are heavily involved in cell therapy clinical trials, as Ben has already outlined. These are just some of the examples that we've been involved in. And as you can see, they are for various different indications. It's not just cancer. We've got some for liver transplantation. We've got some solid organ cancers in there. And then the new ones coming online are sort of in the direction of autoimmunity and lupus. And as Ben said, we've got the first patient being talked to tomorrow in clinic about that. So it is really, really very exciting. And again, in this setting, cell therapy has exactly the same role in the laboratory. Next slide, please. So what's next? So cell therapy lab has undergone some major changes in the past 5 years, and now we are standing on our own 2 feet again, and we are really looking ahead to the future. So the goals I've got for the department are shared by the trust strategy boards. I want to diversify the routine service. We really need to bring some particular processes in-house that we currently outsource, and we do need to increase our capacity as well to meet the clinical demand that's coming from our existing cohort, but that also will come from the new hospitals that are being built on site as well. We want to diversify our portfolio of clinical trials for cell therapy into autoimmunity. So this has already begun with lupus, but we're also looking into myasthenia gravis, multiple sclerosis, and looking at the neurology settings as well, because there are so many unmet needs out there that need addressing. We're also looking to increase the commissioned CAR T service as well, as new products are now coming online onto the market. Two that we're looking at introducing this year. I'm not even going to try and pronounce them, but one of them is for follicular lymphoma and one of them is for multiple myeloma as well. So really diversifying the treatments that we offer. And then what I'm really passionate about, and I think most of you have probably heard of, if you're interested in CAR T, is that we will shortly be expanding to a new facility in the Geoffrey Cheer Biomedical Centre, where my plan is to develop an in-house manufacturing arm of the service. Next slide, please. So the JCBC is a brand spanking new building, just a stone's throw from where we are at the moment. And if you know where car park 4 is, it's next to car park 4. A portion of the 5th floor is earmarked for our laboratory, and the validation of it is already starting and will be, I hope, fully operational within about 18 months' time. The facility will provide us with 2 additional clean room suites, so four rooms in total, divided into 2 suites. One will be reserved for sort of the routine service that we offer at the minute, but the other half, because it's capable of handling genetically modified organisms, it's a perfect setup for being able to manufacture cell therapies, particularly those that involve GMOs such as CAR T cells. The facility also offers wet labs, offices, and storage. So, and the plan is to expand into this space. We're not moving, we're expanding. So this will give us many, many opportunities to diversify the service, but also expand our numbers as well. Next slide, please. So these are some pictures that Ben's put in for me to give you a little snapshot of what the lab looks like. I kind of feel like it's what NASA might look like. It is very space age. It's very shiny and new. It's quite empty at the moment, but after recent financial approval, I've got the go ahead to now purchase a load of new exciting whizzy equipment to go in there. So that will happen very shortly as part of the validation. And the last slide is basically to outline what this expansion to JCBC will enable us to do. So ultimately, it will enable us to deliver more therapies to more patients. And these patients are not going to come just from our existing cohort. They will be coming from the draw of the 2 new hospitals that are being built on site. So we've got the Cambridge Cancer Research Hospital and the Cambridge Children's Hospital, both set to open in 2029. And I need to accelerate the development of the cell therapy service at quite a rapid rate to be able to meet the demands that these 2 hospitals will bring. We want to increase access to the wider of East of England. So as Ben showed in the map, we really do cater for a really huge part of the UK. And we don't want patients from Lowestoft, Kings Lynn, Yarmouth to be going down to London or going up to Manchester. We want to be able to treat them as close to home as possible, and that means Cambridge. The increased lab space will also give us a bigger processing footprint, so we can just do more of what we do already. But particularly, it will give us the opportunity to be able to manufacture cells on site. Much closer to the patient, we don't have the time delay of sending cells all the way across to the States or to Europe to be bothering with customs and things like that. We also will get to manufacture cells within a few hours of them coming out of the patient, so they're nice and fresh, and that really does make a difference in the cell therapy world of manufacture. And it sounds cheesy, but using the amazing academic potential that we've got in Cambridge, we can really slingshot that bench to bedside novel treatments and further research in this area and move closer to becoming a centre of excellence in cell therapy. So with that, we do have a few minutes for questions. So thank you so much for listening. It must be an overwhelming amount of information, but I hope you've all found just at least one thing you can take away that you'll find useful. So thank you very much.
Thank you very much indeed, Sarah and Ben. I can see from the number of clapping hands that people have really found that very interesting. I've got to say, I mean, this is clearly one of the most exciting areas of medicine, and it's just growing at such a rapid rate. So absolutely amazing stuff. There was one question before the, I think we've probably got time for one question, sadly. Other questions, if you can send them in, we will answer those. We'll get back to you with answers. But if you could also complete our short survey, please. And a recording of this webinar will be on the CUH website if you'd like to share it with friends, because I think a lot of people are going to be very, very interested in this area. We've got comments there like outstanding, etc. One question, very quick question, and that's about myositis. Could you comment on that, either of you, please?
Yeah, certainly. So inflammatory myopathies or myositis are one of the areas that are being explored for CAR T-cell therapy and autoimmunity. And at the moment, there are data that I began to show you, but they're quite new. It's a relatively small indication, or rather, the studies into it have been relatively small. So it's clearly an area of interest for a number of different pharmaceutical companies to develop a CAR T cell that could be used for myositis. I'm in conversation with actually several different companies to see if they would be interested in launching a trial with us to treat inflammatory myopathy. And part of the challenge for doing all of these trials is to make sure that we have a dedicated cell therapy team, which of course we do, but also that you have a dedicated team who can treat inflammatory myopathies, as we also do. So we're fortunate in Cambridge to be able to bring together a number of teams with both disease-specific expertise and cell therapy expertise to deliver these kind of treatments.
Thank you very much indeed. I'm not sure if we've got time for any more questions. If you can send questions through, Namug, would you like to comment on how people can send other questions through after the event? Because as I said, there's been so much interest generated. The comments that you've sent back, thank you so much for the lovely comments to the team. They are truly amazing, aren't they? But Namu, over to you.
Sure, thank you so much, Chris. So if you want to send, also I've got your questions here, and also we've got your your email address from when you've registered, so on the database. So we will gather those answers for your questions, and then we will email you the answers. So I'll liaise with both Sarah and Ben, who will help with the answers, and I'll get back to you.
Okay, we've got 2 minutes left. Just a final comment from either of you, but thank you so much. That was truly brilliant. But over to you two for a final comment.
Sarah.
Oh, it's just an honour to be here, and it's not very often that I get the opportunity to really showcase the laboratory side of what goes on in these clinical trials and things. So just thank you for the opportunity, Ben.
And for me, yes, this is clearly an area that I'm absolutely passionate about and have been for many years. We're very fortunate to be working in an area where we can bring this to patients, and we can not only do that, but we can work somewhere like the Cambridge Biomedical Campus, where we have the opportunity potentially to develop globally novel therapies that could then be used to treat patients worldwide. It's a really exciting prospect, and it's fantastic we're able to do it in Cambridge, and I'm delighted to have the opportunity to tell people about it. So thanks very much.
Thank you all for attending, and thank you to our speakers. And I said, if you can remember to complete the survey, that would be great. And this webinar is going to be available. I think it's going to be the most popular one ever. Thank you very much, and good night all.
Thank you so much.
Thank you very much.